Hello, everyone, and welcome to today's AnaBios webinar. My name is Chris Mathis, and I'm the Chief Commercial Officer at AnaBios. Before I introduce today's speaker, I would like to say a few words about AnaBios. AnaBios is a unique contract research organization and biotech company based in San Diego. We recover human organs from ethically consented donors and use the tissue or cells to conduct physiological assays in which we can test preclinical compounds for drug discovery projects. We've essentially redefined first in human. And working with AnaBios as a partner is as close as you can get to a clinical trial without actually doing a clinical trial. This slide shows some of the benefits of AnaBios' novel translational research. So it's my pleasure to introduce our speaker today. Jouko Levijoki is a scientist at Orion Pharma in Finland. And Jouko will be talking today about testing a compound that has implications for heart failure. And we're actually, AnaBios scientists co-authored a publication that came out recently in the British Journal of Pharmacology. And AnaBios is announcing this in a press release in about an hour from now. So Jouko has been at Orion Pharma for about 30 years, and he's been quite busy during that time. He has 14 patents. He's been in over 40 publications, and he did his master's at the Helsinki University, also in Finland. So Jouko, if you could go to the next slide, please. It's my pleasure to introduce our speaker today. Could you go to the previous slide, Jouko? I just want to read the title.
Yes.
The title of the presentation today is Discovery and Characterization of ORM-11372, Implications for Heart Failure Drug Discovery. Jouko, welcome to the webinar, and take it away, please.
Okay, thank you very much for introduction and opportunity to discuss about how we ended up to this molecule. So everything started something like 2000 around, and before that we involved to many cardiovascular projects like levosimendan and others molecules which were interacting with calcium handling proteins. So NCX is quite difficult target to study due to its dynamic nature, but we know that it has involved to many things like arrhythmias. It is definitely behind of heart failure in some extent. Very well known situation is when we have a ischemia reperfusion. So ischemia reperfusion injury is mediated via NCX. It has also part in blood pressure regulation and many other things, including cerebral ischemia, stroke, and even neurodegenerative diseases. Something about the NCX structure. It has 10 transmembrane helixes, which are organized in 2 clusters. And between those 2 clusters, there is this regulatory loop, which contains calcium binding sites. And NCX has 1 calcium binding for calcium and 3 for sodium. NCX has 3, let's say, the major subtypes, and NCX1 is distributed all over the body, so it makes it quite hard to attack specifically. NCX2 and 3 are expressed in brain and skeletal muscles. In addition to these major subtypes, there is let's say the splice variants, and all splice variants are derived from this second calcium binding domain. Different splice variants arise from different exons, and exon A is connected to excitable tissues like the heart, skeletal muscle, neurons, and B to non-excitable tissues like the kidney. We know that there is at least 17 subtypes for NCX1, none for type 2, and at least 5 for NCX3. And NCX is like a wind castle. Its direction depends on sodium concentration above the membrane as well as calcium concentrations. And in addition, that membrane potential has impact to this equation. NCX exchanges uneven number of charges over the membrane, so it is electrogenic. And depending on its mode, in forward mode, it will induce the depolarizing current, and in the same time it will extrude calcium, and in reverse mode it introduces calcium into the cell and causes hyperpolarization. Here is the sketch how we were organized in this project. So there was first the screening and synthesis and optimizing of physical chemical properties, and those molecules which were potent enough were going to the second level screening. And we knew that most difficult or we should have a good selectivity between the NCX inhibition and calcium channel inhibition. So that was the, let's say, the major tasks at the beginning. I said our development started around 2000, and at the time there was only known small molecule structure was the AnaBios compound KBr 7943. And meanwhile, we were developing our own compounds. SEA compound structure was published, and that was incorporated in our molecular modeling. So based on these known structures, we ended up the idea that with making a molecule scaffold more stiff, we will get a better potency. And here we were sketching the possible modifications for the existing NCX inhibitors. And the first, our own compound was the 1010-3. Although this 1010-3 was more potent than CA400, it was still quite poorly soluble. The next step in this development was to improve the solubility, and we discovered that from the other end of molecule, it was possible to add amino acid like structures which improved the solubility from microgram per milliliter to milligram per milliliter level. And that make it possible to study these molecules also in vivo models. Real big advance happened between this NCX A series to B series. So based on this A series, we built up the model which utilized all the good features of this AA series, and we made a virtual screening campaign, and from that we ended up to hit molecule 120407. That was already had a quite good NCX inhibition, although its selectivity towards L-type channel was still quite lousy. And based on this optimization, we could make a pharmacophore, which is shown here. So here are the critical elements which are needed for NCX inhibition. And now we are jumping to actually the results. So with this optimization process, we ended up this 11372, which was the let's say at the moment the best most potent molecule we had. The screening was done in SF9 cells using fluorescence detection, and that facilitated many molecules to screen at the moment at the time. And what we see here is the human inducible pluripotent cardiomyocytes, and this is NCX in current, which is measured by patch clamp. As we can see, this inhibition of NCX inward and outward current are exactly the same. The IC50 is around 5 nanomolar. And in order to know that what are the species differences, NCX Rat NCX was studied in rat primary cardiomyocytes, and the result of this is that NCX inhibition pretty much the same level. It was something like 10 nanomolar, and also we didn't find any unidirectional block, or let's say the blocking was same for inward and outward currents. Okay, to sum those electrophysiological and calcium measurement studies, here's the summed effect in human NCX current versus other cardiac currents. In this figure, it's easy to say that we have a good selectivity, and especially towards L-type calcium channel. So it's something like 1,200 times selectivity. Then coming to the indications. So one of the indications was improvement of cardiac contractility. In vitro, we can see that NCX inhibitor can induce increase of contractility. This is effect in guinea pig papillary muscle, and same phenomenon can be seen in vivo studies. Here is some effects in normal rats, anesthetized rats. So with the increase in infusions, we will see the marked inotropic effect, and also study what is the effect in diseased states. We used infarction rats, where we can see that this, let's say, the base level contraction is reduced markedly because of infarction, but still we can induce inotropy with the NCX inhibitor. We also were interested to see what is the species differences, and here is shown also effects in anesthetized rabbits, and we can see that there is also increase in heart contractility. At the beginning, we were quite worried about that what is the effect on relaxation if NCX is inhibited, because it was very well known that the NCX is one of the major calcium extruder in calcium handling. So it was we were surprised that actually we didn't find any weakening of relaxation here in left side panel. This is once again guinea pig papillary muscle. So here is vehicle and here is the active compound. So no effects on relaxation. In vivo, we also find no worsening of relaxation. It was remained the same. Only what was very surprising that in infarct rats at very low concentrations there was amazing improvement of relaxation at the lowest concentration. Other aspect of cardiovascular effects, one is related to heart rate control. It is very recently published review about that by Koheda et al. And the main message was that the basal heart rate is dependent on pacemaker current, and NCX current is modifying the heart rate, like when there is need in flight or fight situations. And basically 20% remaining of 20% of NCX current is enough to maintain the normal heart rate. So here is what we saw in vivo. Once again, we didn't see so much in normal rats or rabbits. However, in myocardial infarction rat, there seems to be dose-dependent decrease of heart rate. So there might be change in the control of NCX or the other ion current. Okay, then we are coming to the safety issues, and NCX current in normal situation is let's say that it is quite minor, or it is in forward mode. So if NCX current is inhibited, it should shorten the action potential duration. However, in this left panel, we can see that in human cardio ventricular tissue, trabeculae, we couldn't see any effect on action potential duration. That this was contrary to what was seen in single IPS cardiomyocytes, where quite markable shortening was seen. If we compare species differences, it seems to be that we don't see action potential shortening in guinea pig papillary muscle, dog Purkinje fiber, or human trabeculae. And these concentrations were very high in dogs at very high concentration, like 30 micromolar. We will see shortening action potential, but this is probably due to loss of selectivity and could be due to L-type calcium channel inhibition. And further, this safety assays, NCX inhibitor didn't show any pro-arrhythmic signs in human trabeculae, and this was confirmed in dog Purkinje fiber model. So there was no early after depolarizations or delayed after depolarizations. So no signs of it could cause dorsal dead points or definitely not good interval prolongation. And actually for this publication, we was asked to make a confirmatory study. So it was done in hex cells where human NCX1 was expressed, and in this model, NCX is operating in forward model. So in head-to-head comparison, it can be seen that 11372 is the most potent known inhibitor, and in comparison of 1010-3 or SEA or KBR compound. And here is the comparison with other inotropes. This figure is from guinea pig papillary muscle model, and we can see that NCX inhibitor is as potent as levosimendan, and the same scale with diabutamine or isoprenaline. And we also made a pharmacokinetic dynamic comparison with dobutamine, and in these figures we can be seen that when these plasma concentrations are converted to the free plasma concentrations with 11372, we can see the linear increase in contractility, and starting even lower concentrations than dobutamine. And heart rate profile is of course different. So dobutamine increases heart rate quite much, and NCX inhibitor have a slight decrease in heart rate at the very high free plasma concentrations. So based on this exercise, we can conclude that 11372 is first third generation NCX inhibitor, and it is probably the most potent and selective inhibitor what is reported at the moment. It has a good selectivity towards other ion currents, and we show that it can induce positive inotropic effect also in rabbits, which has been before not shown. And its hemodynamic profile differs from the, let's say, the old school inotropes. That's sure. So no increase in heart rate and no decrease in blood pressure. And of course, there's something to study more. So with these low concentrations, there was this unexplained improvement of relaxation in function model, and one very important point was that the safety profile of NCX inhibitor is non-pro-arrhythmic. So this is the, let's say, the overall aim what we can do in cardiovascular medicine. So okay, thank you. That's all from my side.
Thanks very much, Jouko, for an excellent presentation. For those of you in attendance, you can ask questions now by writing them in the question box. You'll see that on the GoToWebinar dashboard towards the bottom. And while people are entering questions, Jouko, I have a question for you. So, you know, drug discovery teams in biotech and pharma are really working hard to deliver the next heart failure therapy. This next therapy could be developed using small molecules like the compound you described today, cell therapy, or cardiac regeneration. Do you think these strategies have equal chance, or do you think one can be better than the other?
I think that this kind of compound could introduce a new modality in the patients who have a low blood pressure or don't stand for the increase in heart rate. And my guess is that it's could be combined with the existing medication, so improve the efficacy in inotrope non-tolerant patients.
Okay, thank you. Here's a question from someone in the audience. Is ORM-11372 having a longer PK than other NCX inhibitors?
Excuse me, could you repeat?
Yes. Is ORM-11372 having a longer PK than other NCX inhibitors?
No, it is shorter. So it's our aim was to have like a on-off inotrope. So with using infusion rate or speeding up the infusion rate to get the more efficacy, and if we need to reduce it, just to reduce the infusion rate. So that was the like easy to modify the effect, okay. like a on-off inotrope. That was the aim. So for acute use, I would say.
Okay, thanks. Yeah, once again, if you have a question, you can enter it in the question box at the GoToWebinar dashboard. I found it very interesting, the safety slide you showed, where you showed the AnaBios human cardiomyocyte data, where there was no change in action potential duration. But in the stem cell cardiomyocytes was. What do you attribute that to? Do you think that has to do with stem cells just having the immature phenotype and not expressing all of the proteins that adult human primary myocytes have? Yeah, this slide.
Yeah, good question. Yeah, that might be one of the points that these current densities are not exactly same as in major mature cardiomyocytes. And one thing what might be behind this differences is that this single cell is in huge volume, and so basically ion concentrations outside the membrane are diluted to this huge volume. So I think that maybe this isolated cardiomyocyte, I think that they are maybe overestimating the effect. So surely there is this effect that it, like here in NCX, the action potential duration will shorten, but with this single cells, it is more prone to happen.
Interesting, thanks. So here's another question for you. Reliable human cardiac data from normal and heart failure organ donor hearts can now be generated at the preclinical stages of drug discovery. How do you think such human cardiac models can be integrated so efficacious and safe novel therapies can be progressed to clinical evaluation? So in other words, you know, the human cells and tissues that AnaBios works with, how do you see that as being part of your drug discovery program?
That's a very huge advance what you are doing. So as seen in this slide, there is a different species, and of course, when you can test the compound in human tissue, it is you don't need to this species to species evaluation. So you have a very good idea that what will happen when you are developing the human drugs. So I think that's the excellent.
Yeah, it's all about the translation, right?
Right.
Yeah. Okay. Well, I think this case study was an excellent example of how AnaBios collaborates with partners, and we really appreciated the partnership with you and Orion and your team there. As I mentioned at the beginning, those of you in the audience that are interested in reading the paper in full, you can download this from the British Journal of Pharmacology or from the AnaBios website. So here's another question from the audience. So heart failure programs have so far focused on direct measurement of contractility in a variety of models, but the translation into clinical efficacy has been disappointing so far. What do you think is missing, and what are the other endpoints you think would be relevant?
Yeah, that's a tough question. It's I think that has been always the problem, that what you see in acute situation, that does it really have a positive impact on survival, and I think that that's needs to disease modifying drugs, and that's most probably with inotropes we can just have a time to other modalities to work or to keep things ongoing. But I think that there's this kind of like if you can turn this heart failure remodeling back, then it's I think that's what is needed, and
right.
we are still looking for that kind of treatment.
Okay. Here's another question from the audience. So this drug that you're describing, this compound or others like it, would it be used for acute heart failure, or would it not be used for long-term usage?
This one is for acute.
Acute, okay. And do you have myocardial infarction model efficacy data for this compound?
Do you mean like a acute ischemia situation?
Right. Mm-hmm.
Not sure for this, because those infarction models, they were like 10 days later after the infarction. So we were looking for the decompensated state, and then to see, so all these damages were already happened. But surely this acute like ischemia reperfusion injury is one of the where we can have an effect with this NCX inhibition. That's a
Okay. All right. Well, once again, thank you for an excellent presentation. Thanks for a great collaboration. And those of you in the audience, thanks for joining us. Be on the lookout for a future AnaBios webinar. We have some scheduled in November that we'll be announcing soon. And thanks everyone for your attendance. And we look forward to having you back in the future. Thanks again, Jouko.
Okay, thank you for your support and good collaboration. Yes.
Thank you.
Okay, bye-bye.